Hello, my name is Evan Watchman, and I'm going to be going over cell biology. Okay, 1.1 Introduction to Cells. Um, Cells are basically the smallest units of, according to cell theory, cells are the smallest units of life. They come from pre-existing cells and are the building blocks to all living organisms. The cell theory gives an explanation for the relationship between cells and living organisms, and also we can thank the development of the cell theory to advancements in microscopy, as they were able to see what the human eye couldn't. Live micros, for example, the invention of the light microscope confirmed that animal and plant cells were made up of individual cells, which further proved the point of the cell theory. And the electron microscope allowed us to see more specific internal fine structures in a cell. To calculate the magnification of these items of these microscopic um, cellular cells, we have a formula which is the size of the drawing versus the actual size. So we basically just divide the size of the drawing that we have versus by its actual size to get the magnification. So if I were to have a drawing that measured about 10 centimeters long, with the actual size of the cell being 100 micrometers. If we were to do 10 centimeters divided by 100 micrometers and convert 10 centimeters into 100,000 micrometers, that would basically mean 100,000 divided by 100, which is 1,000 times magnification. All living things have 7 crucial life functions: metabolism, growth, response, homeostasis, nutrition, reproduction, and excretion. Without all these 7 things, uh, a thing cannot be considered alive. An example of this is a virus. Um, Examples of unicellular organisms are the Paramencium, which is a unicellular protozoo, and the Chlamydomonas, which is a unicellular green algae. The reason why we can't see cells is because they're prevented from growing too large and instead have to resort to dividing into smaller cells because of something called surface area to volume ratio, which basically means that it kind of determines if a cell has good efficiency or not because of the surface to volume ratio. And a lower volume surface area to volume ratio makes a cell more inefficient because it has to travel a much larger distance to transfer uh, like materials or nutrients. And so growing too large makes the surface area to volume ratio smaller, so cells divide to have larger surface area to volume, which leads to better efficiency in cell functions. We also have multicellular organisms, which had developed differentiation in their cells to perform different tasks more efficiently. This caused multicellular organisms to display emergent properties because of the many things it could do that single cell organisms couldn't because of cell differentiation. We also have stem cells, which are undifferentiated cells that have the ability to divide indefinitely, which means it's able to divide up until uh, its decease. And the offspring of these cells have the ability to cause differentiation. So it's able to perform a task in a specific area for a specific area. 1.2 Cell biology. About 550 million years ago, multicellular life first appeared, evolving from unicellular organisms that had begun to work together. They encountered a problem in their inefficiency to transfer energy between cells and the environment. So to help solve this, um, a cell differentiation was created, which allowed cells to specialize in different functions and this created a range of evolved specialized tissues in their bodies. The two most generic evolutions of multicellular life are the prokaryotic cell and the eukaryotic cell. The prokaryotic cell is considered the earliest and most primitive cell that originated 3.5 billion years ago. This cell includes bacteria and archaea. The prokaryotic cell has no nucleus or membrane-bound organelles, with the metabolic process occurring in the cytoplasm. Meanwhile, the eukaryotic cell are complex cells with genetic material in the cytoplasm by the nucleus. The cell isolates itself from certain parts of the cell from the rest of the organelle to help increase metabolic efficiency, isolate toxic or damaging substances, and to give it the flexibility to change its numbers and position of organelles within the cell. Okay, 1.3 membrane structure. The structure of a membrane is something called a phospholipid bilayer, which is basically a lipid with a phosphate group that consists of two layers of phospholipids that are arranged according to their properties, which are amphipathic properties because of the phosphate head being hydrophilic, while the fatty acid head is a hydrophobic. Shown within the fluid mosaic model, which is to the right in the picture, we can kind of see that the little blue circles are the phosphate heads, which are hydrophilic, which means that it is used to the water, and the fatty acid tails, which are hydrophobic, which it doesn't like the water, so it's turned inwards. The fatty acids are turned inwards to each other, so they don't have to deal with the aquatic environment. This model replaced the Davison-Daniell membrane model because of this older model's shortcomings. That was assumed that all membranes had the same structure, which did not explain how different types of membrane could perform different functions, or how most of the proteins were mostly hydrophobic, which would have made impossible to have an aquatic environment on either side of the membrane. For cholesterol is a steroid of nonpolar comprising 4 ring structures. It also is made up with a hydrocarbon tail and a polar hydroxide group, which is also hydrophilic. It can enter into the cell membrane due to its empathic properties. 1.4 membrane transport. We have two different types of transport: passive transport and active transport. Passive transport is classified into two things: diffusion and osmosis. Diffusion is the movement of particles from a high concentration to a low one, and within diffusion there are two types of diffusion: simple and facilitated diffusion, which are both affected by various factors such as temperature, surface area of membrane, size of the particles, and concentration gradient of diffusing particles. And then we have osmosis, which is the passive movement of water molecules from regions of low concentration to regions of high concentration. So it's the exact opposite of diffusion. And this only involves water molecules, with the solvents that are the guides that lead the water towards whichever side has a higher concentration of solvents. And osmolarity is the concentration of a solution in terms of moles or sols per liter of solution. And then we have active transport, which requires energy in the form of ATP to move particles across membranes. It uses to transport substances. It uses active transport to transport substances from regions of low concentration to high concentration. And we can visualize this model as a sort of like a sodium-potassium pump. Endocytosis is a cell process where substances are taken in from the outside environment and consumed by the cell. And there are two types of endocytosis depending on what the cell consumes. We have pintocytosis, which is consuming liquid substances, and then we have phagocytosis, which is the absorption of solids. And then apart from endocytosis, we have exocytosis, which um, ejects materials from the cells. And this can be classified as two processes, excretion and secretion. Excretion is just a process where it excretes indigestible parts of the cell that are not useful, so it basically just gets rid of waste and other things that just wouldn't help the cell. And then we have secretion, which is proteins that are synthesized by ribosomes and first pass through vesicles where uh, they are processed before being released in vesicles that fuse with the plasma membrane for secretion outside the cell. And vesicles allow for the movement of materials in cells. 1.5. The origin of cells. It has been debated for a while about where the first origin of cells were, with an idea of the theory of spontaneous generation, which basically said that cells appeared out of the air, and to try and experiment with that, Louis Pasteur made an experiment which basically disproved this theory of spontaneous generation and further supported the theory that cells came from pre-existing cells. And with the Miller-Urey experiment, it further proved the existence of the first ever cell, as it helped to understand what was necessary for life to occur, with 4 conditions believed to be needed for life: simple organic molecules, larger organic molecules assembled from smaller molecules, reproduction of organisms, and self-contained structures that are required for biochemical reactions. The origin of the eukaryotic cell is called the endosymbiotic theory, which explains how the mitochondria and chloroplasts were once long ago themselves prokaryotic beings that were consumed by a larger prokaryotic cell. But instead of being digested, they remained inside the host cell and performed different tasks for that cell. And this theory can be backed up by the fact that the mitochondria and chloroplasts both have double membranes, both have circular naked DNAs, they both have DNA formed as single chromosomes, they both have 70S ribosomes, and the ability to divide by binary fission, and they both are vulnerable to some antibiotics. 1.6 Cell division. Basically, cell division, this is the cell cycle, and the cell cycle can be divided into 3 stages: interphase, mitosis, and cytokinesis. Interphase is the longest and most active time of the cell cycle, with many of the cell processes occurring here, like metabolism, endocytosis, and exocytosis. Interphase encompasses 3 important phases of the cell cycle, which is G1, S phase, and G2. And then after interphase, we have mitosis, which is where the division of the nucleus begins and splits into 2 identical daughter nuclei. The 4 phases involved with mitosis are prophase, metaphase, anaphase, and telophase. And then after metaphase has occurred, after mitosis has occurred, we have cytokinesis, which states that it divides the cytoplasm of the parent cell between the 2 daughter cells. Cytokinesis is different for both animals and plants because of the presence of the cell walls in plant cells, which is shown on the right as animal cells split apart while plant cells just kind of stay together, but like separate but not separate. Like they're both attached, they just split apart a little bit more. And cyclins are proteins that manage the growth during the cell cycle. Cells cannot reach the next phase unless cyclins reach a certain level of concentration, which once it reaches that concentration, it binds to enzymes called cyclin-dependent kinases to activate them, or CDKs to activate them. The CDKs then attach to phosphate groups to other proteins in the cell, which then triggers other proteins to become active and carry out tasks that are specific to a certain phase of the cell cycle. 1.6 continued. Tumorigenesis. Tumorigenesis is the formation of a tumor, which is basically a mass of uncontrolled replicating cells. There are two types of tumors. There's a benign tumor, which is usually limited to a certain area and doesn't really spread to other parts of the body. Most of these tumors are treatable. And then we have a malignant tumor, which is a cancerous tumor that can be resistant to treatments and might be able to spread to other parts of the body, and even when removed can reoccur after being treated. These tumors can be caused by mutations in a cell that loses its ability to enter to the G1 phase and instead just multiplies uncontrollably. Mutations are caused by changes in the genetic code, and gene mutations are caused by mutagens. And after genes in a cell have undergone a mutation, oncogenes we get oncogenes, which also contribute to the production of a tumor. An example of tumorigenesis is smoking. Smoking can increase the risk of many different types of cancers and other diseases. Okay, thank you for taking your time to listen to me.